Hi, can you hear me?
Yeah, I can hear you, Sitabhra. Yeah.
Yeah, something suddenly happened. Everything just
I
went
don't know
off.
what happened. I think everything.
Was my connection lost? Was my introduction lost?
I
Yes.
think everybody kind of, went off in orbit for a few moments.
Oh
Yeah,
God.
same.
We lost the because I was on a different screen. I did actually was looking at it. So should I?
Yeah, Shunatra, can you please restart?
Okay, but I think I've lost the presentations window.
Yeah, can you just allow me to share content once more? Because I think I've lost that right, probably.
Okay. Do you?
Yeah, great. Thanks.
Yeah, we are able to see now.
So Shuneta,
Yeah, I will just give the intro once again. I'm so sorry.
sure, no problem.
Yeah, so our next speaker is Professor Sitabhra Sinha. He is a member of the faculty in the physics group of Institute of Mathematical Sciences, Chennai, and an adjunct faculty of the National Institute of Advanced Studies, Bangalore. He did his PhD at the Machine Intelligence Unit, Indian Statistical Institute, Calcutta, on the nonlinear dynamics of recurrent neural network models. He did his postdoctoral research at the Department of Physics, Indian Institute of Science, Bangalore, and Vail Medical College of Cornell University, New York, focusing on nonlinear dynamics of spatially extended systems with application to biological systems. He joined IMSc in 2002. His research interests are broadly in the areas of complex systems, nonlinear dynamics, and theoretical and computational biophysics. As I said before that, his talk title is very interesting. And Shan was, we were discussing, Shan said it's William Blake. The title of his talk is Framing the Fearful Symmetry, Developmental Pattern Formation as Interaction between Global Fields and Local Interaction Among Cells. So before I lose my connection, over to you, Professor Sinha.
Thank you so much, Sunitra, for the introduction, and also all the organizers of the workshop for asking me to talk about our recent work. So you're, of course, absolutely right. The title of my talk is inspired by the famous poem of William Blake, whose last few lines goes as follows. Tiger, tiger, burning bright in the forests of the night, what immortal hand or eye dare frame thy fearful symmetry? So the reason I picked this particular uh, quotation to introduce my talk is because there's a recent twist to this whole idea of a fearful symmetry. So Jan Stewart, who's a very famous mathematician and math popularizer, in the 1990s, he wrote this wonderful book with Martin Golubitsky called Fearful Symmetry. And the beginning of the book, they actually refer to Blake's poem and say that, look, in the modern context, we have a different interpretation of this poem. So, I'm quoting directly from Stewart. He says, the fearful symmetry of the tiger lies in the dynamics of biological development, which creates a miracle of striped perfection from a spherical egg less than a millimeter across. Fearful indeed, but in the sense of awe-inspiring. And if you have seen, any of the recent videos, which are available in many places on the internet, which shows how every organism on this planet starts life as a single cell. But that single cell, through a sequence of cell divisions and differentiations, eventually become a particular organism. It could be the tiger, it could be the mouse, it could be a human being. So, unfortunately for human beings, we still don't have an actual, video, like time-lapse video showing how you have a single cell essentially developing into a human embryo. I guess for ethical reasons, you can't really do such a video, but we can do it for many other organisms. And one of the most spectacular videos is that of a zebrafish zygote, essentially a fertilized egg, which by definition is a single cell, how over the course of just 85 hours goes from a single cell to essentially a recognizable zebrafish embryo. So let me just show you the video. Hopefully you can see it, right? Are you able to see the video?
Yes.
Okay. So at the bottom right corner, you see the time that has elapsed. And essentially, the beginning of the video, it was just a single fertilized cell. From that, you have the cell dividing. So initially, one cell divides to 2, 2 to 4, 4 to 8, 8 to 16, 16 to 32. And initially, each of the cells are completely identical. So there is complete symmetry, complete homogeneity. But after a while, different cells start behaving differently. And within about 20 hours, you see a recognizable organism with a tail, with a head. And you can see essentially the somites forming. More about that at the end of my talk, which essentially would form the backbone. So you have the tail elongating. So, once you have enough differentiation, once you have, all the specializations happened, the brain has formed and so on, you essentially have a growth of the organism and eventually something which, quite clearly looks like the embryo of a fish develops. It's a marvel that starting from something which was completely undifferentiated, which was just like an undifferentiated collection of identical cells, you have something which has a clear form, something which is extremely characteristic of a particular species. The same thing holds true for every other living organism on this planet. And so the question is, how does this happen? Now, of course, a biologist would be very interested in the explicit mechanisms. And so, for example, one biologist might be interested in how this series of steps happen for humans. Another could be interested in how this happens for, let's say, the zebrafish. Another could be interested in how this happens for, let's say, the Sinohabditis elegans, which is, one of the favorite model organisms for people studying multicellular creatures. But for a physicist, in a sense, all of this reduces to just a single question. How are the symmetries that are there in the initial state broken? How do you have what is essentially a completely homogeneous entity developing into this, very heterogeneous, very asymmetrical kind of system? Is there anything that physics can offer to understanding this puzzle? And in that sense, I'm going to talk about, the symmetry of the organism and how you break it to actually get the recognizable shape of the organism that we are interested in. Now, if you look at what present day biological theory has to say about it, if you, for example, go to a classic text in developmental biology, for example, that written by Wolpert, or for that matter, any of the other several very good textbooks in developmental biology, you'd see that the usual answer is framed in terms of the concept of positional information. So what is positional information? Essentially, it is any signal that allows the cell to recognize where it is located with respect to the other cells within the system. You have to have some kind of GPS, so to say. Of course, cells don't have GPS, but you have to have something akin to GPS so that the cell knows where it is. What is its relative location within the assembly of all other cells? And knowing this, knowing where it is, it can actually decide to do certain things which other cells will not do. So every cell, based on information about where it is located within the body frame, so to say, decides to go along a particular program. Now, what is surprising is that every cell actually has the same genome. Every cell has exactly the same DNA. However, by regulating the expression of the genes, you can actually allow certain genes to be turned on and other genes to be turned off in specific cells. And so therefore, certain cells will, depending upon where it is located in the body, become brain cells. Other cells will become liver cells. Other cells will become muscle cells, and so on. Now, the interesting part is that the mechanism by which a cell gets to know where it is and it's the program that it initiates once it figures out where it is, these two need not be related. And so you could have the same process by which you are getting information about your position operating in different cells, but every cell, upon knowing where it is, could actually embark on a completely different program, giving rise to a different cell type. So Wolpert, who's actually one of the pioneers in this area, gives a wonderful example of this. So he says, imagine a large stadium where the spectators are given colored boards of different types. And depending upon where they are seated in the stadium, they are given different boards. Now, at a particular time, the spectators are instructed to hold up a particular color board. So when each of the spectators holds up her assigned color board, you could have a pattern emerging across the stadium. For example, here we see, where each of these individual spectators are holding up a colored board, and the end result is something which spells out the word goal. So if you have some way of telling every cell where it is, so essentially the seating plan of the stadium is, given to each of the cells, so, the cell knows where to go and sit, then the particular program that it embarks upon would be essentially equivalent to, like, what are the colored cards that it has been given and the instructions it has been given about, when to hold up each colored card. So the question is then, how do you actually give the cells this positional information? How do you tell the cell, look, you are located in the head, or look, you are located in the tail? So this is where actually, kind of the controversy, so to say, arises, because currently, at least in the biological developmental community, developmental biology community, I'm sorry, the dominant paradigm holds that this information is given by gradient of one or several chemicals, which are collectively known by the name morphogen. So this idea essentially was pioneered by Lewis Wolpert in the late 1960s. So although it is currently the dominant paradigm, in the late 60s when Wolpert first proposed this idea, actually it was not very well accepted, and he had to struggle against a lot of disbelief before eventually experimental findings seemed to suggest that the picture he was proposing was correct after all. So Wolpert used the analogy of a flag, in particular the French tricolor, to describe how you could have different cells going to different fates depending upon where they're located, using essentially a diffusing chemical, which would have, because of the very nature of diffusion, different concentrations at different locations. So the idea that Wolpert proposed was that, imagine that you have a particular chemical whose source is located at the left of a particular domain. Now, as the molecules diffuse through the domain, you have the development of a concentration gradient, which of course would have the highest concentration at the left end and the lowest concentration at the right end. Now, if each cell decides to switch certain genes on or off, depending upon the concentration of the chemical, then you could actually, in principle, work out a mechanism by which cells which are located closest to the source of the chemical would, after seeing a very high concentration of the chemical, switch on certain genes and switch off other genes, which will essentially take them to a so-called blue color fate. So these cells are colored blue to indicate that they essentially have a blue fate. Cells which are located furthest from the chemical source, on the other hand, is going to see the lowest concentration of the morphogen. And in turn, they will turn off and on certain other genes, which would make them go to the red cell fate or the red fate. So the cells which are located at the right end essentially would go to the red fate. Cells which are somewhere in the middle would essentially be neither have seen extremely large concentrations, nor would it be seeing extremely low concentration. And so in principle, they can go to a red third fate, switching genes on and off, which correspond to, let's say, a white color fate. So we call it the white fate. And so you have essentially what was previously a completely homogeneous or undifferentiated system developing into a heterogeneous system comprising blue fate cells located at the left, white fate cells located in the middle, and red fate cells located at the right. And because this essentially recreates the very characteristic blue, white, red pattern of the French tricolor, Wolpert actually called his model the French flag model. And this is the interesting part. So Wolpert is essentially saying that, look, a French tricolor is recognizably the flag of France, regardless of whether you see it in a stamp or whether you see it, projected on the wall of a 20-story multi-story building. It's the pattern of consecutive stripes of blue, white, and red, which essentially marks the flag. The size, the absolute size of those domains don't matter. It's only the relative placement of these stripes of color which are the relevant quantity. And so the question is, what kind of mechanism can give rise to it? And Wolpert's answer was that if you have a source of diffusing morphogen, you can actually, depending on, so if you have a large flag, you need a very strong source of the morphogen such that, it is going to diffuse to that larger domain. On the other hand, if you have a smaller domain, maybe you actually use a much lower concentration and so on. And by suitably scaling, you can actually generate French flags which can fit any arbitrary scale of the domain, so to say. Now, this idea became very inspiring to developmental biologists in the 70s when people first experimentally confirmed the existence of morphogens. And very soon, people realized that you can actually use multiple morphogens to create a host of extremely complex patterns. So using this, somewhat of a cartoon image, if you have a single morphogen, then that morphogen gradient can either create a set of horizontal stripes or a set of vertical stripes. But if you have two morphogens, then by setting up cross gradients, you can actually set up far more complex patterns, like, for example, the flag of Norway, or, for example, maybe the flag of the United Kingdom. But is that the only way to generate the patterns that we see in living organisms? Or is it possible that there are other kinds of patterns that occur in the course of development which cannot be simply explained by just positing more and more morphogen gradients? It turns out that there is at least one other competing theory for explaining how patterns can form, which actually appeals to a completely different idea. The idea of self-organization through interacting morphogens. Now, the idea of Wolpert, that of diffusing morphogen, which essentially sets up a gradient, is often framed as boundary organized pattern formation. That's because positional information essentially tells you where you are in the domain with respect to the bounds of the system. So the boundary of the system essentially tells you, in a sense, where you are located with respect to it. In self-organized pattern formation, on the other hand, it's the interactions that take place locally between neighbors which gives rise to heterogeneity, which gives rise to the breaking of the symmetry. The boundary does not play any particular role. And so, in some sense, the positional information that is implicit in the idea of Wolpert's French flag is absent in the concept of the self-organized pattern formation. So, what is the self-organized pattern formation? This goes back to the idea of Alan Turing, who is of course famous as one of the fathers of the modern digital computer. So towards the end of his life, Turing became very interested in the idea of morphogenesis, how form develops in living organisms. His interest in this was, of course, driven by several phenomena that we see all around us in nature. For example, the very characteristic arrangement of florets within a sunflower, which it turns out actually follows the Fibonacci sequence. And Turing was actually extremely interested to see, if there could be some simple explanation of how this mathematical pattern actually develops. But more to the point, he was interested about this question of, how when you have the development of, let's say, a frog embryo, initially, the cell kind of keeps dividing and forms, something like a spherical mass of cells. But at some point, you have a hole developing in this embryo, which would essentially, lead to the formation of the inside and outside of the body, so to say, the process of gastrulation. And so he was very interested in how exactly this symmetry breaking happens. For all external purposes, it seems to be appearing spontaneously. And so the question was, is there a very simple mechanism by which you could have spontaneous symmetry breaking in what is otherwise a completely homogeneous system? So in 1952, Turing published in the Philosophical Transactions of Royal Society this pioneering paper, Chemical Basis of Morphogenesis, in which it posited that there indeed exists a very simple mechanism, which just involves the existence of two chemicals, one of which he called the activator and the other he called the inhibitor. So the activator, as its name implies, promotes creation of its own, promotes its further creation, but it also promotes the creation of the second chemical, the inhibitor. So the inhibitor, as its name implies, inhibits the further production of the activator. Now, left to itself, or left to themselves, of course, this would lead to, some kind of a steady state. But now Turing said that if you allow these two chemicals to diffuse in space, then provided the inhibitor diffuses much faster than the activator, so in other words, you have local activation coupled with lateral inhibition. Lateral because, you're inhibiting your neighbors, so to say. So if you have local activation with lateral inhibition, you could see the spontaneous formation of patterns. In other words, what initially starts out as a completely homogeneous, set of entities would quickly develop into a pattern where some units would be at a high concentration of the activator, some units would be at a very low concentration of the activator. So this juxtaposition of high concentration and low concentration would essentially be manifested as some kind of a spatial pattern. So to understand how this process works, Turing, of course, developed a very complex mathematical theory, but he also gave a very appealing, intuitive explanation. So let me just use Turing's own, intuitive explanation to see, how this process works. So Turing said that, look, you can understand how these patterns form by just imagining an island which is populated by cannibals and missionaries. So the entire island population, either falls into the population of cannibals or falls into the population of missionaries. Now, the missionaries are, all celibate. So, imagine Catholic missionaries. And without any pun intended, we call them inhibitors. So, because they're celibate, the only way that they can keep or maintain their population, because of course after some time they will die. So the only way to maintain the population is to regularly recruit new missionaries from the population of the cannibals. So the cannibals are the activators here. So they of course also die, but unlike missionaries, they can reproduce, and so therefore they can keep increasing the population. So that's how individually each of these chemicals, so to say, work. Now, what happens when you have interaction between these two chemicals? What happens when these two morphogens interact? So that's the rule of the reaction. So the reaction rule is very simple. When two missionaries meet a cannibal, the cannibal is converted to missionary status. What happens when two cannibals meet a missionary? Turing is very coy about, but you can probably imagine the consequences. Now, when the two populations are mixed together, so following usual practice, you mix, shake well, and let it rest. So what will happen is, if you just allow the two populations to mix thoroughly, would essentially end up eventually with a steady state. So some kind of a stable balance between reproduction and conversion is achieved, so that whatever excess of cannibals there might be at any point of time is quickly converted to missionary status. Any deficit in missionary status or any deficit in the missionary population is quickly met by some rapid conversions from the cannibal population and so on. So if you perturb the stable state, essentially after a few perturbations, after a short time, eventually it again goes back to the same original steady state. Now, Turing said, imagine this is taking place in some kind of an island where there's this big volcano in the middle of the island, so that it's completely uninhabited. So the only habitation is, around the periphery of the island, the beach. So, like if you are a fan of, all those musicals of the 1950s, which Hollywood churned out regularly, I'm sure you have seen, these beautiful South Pacific ideals, right? Where typically there would be a volcano in the middle of the island, and there would be this, very Caucasian-looking islanders who are living on the beach. So they're living the good life by any US, standards. And typically, the end of the film would, be, the volcano suddenly, erupting and everybody dying. But of course, our hero and heroine would, eventually, escape using a convenient canoe. So imagine, you are on an island like that, and every cannibal and missionary is kind of limited in how far they can go within a given time, by the speed of walking. So, like they can typically only walk to their neighborhoods. So, they are in some sense diffusing to their neighborhood, but both missionaries and cannibals are doing it at the same rate. So the diffusion rates are exactly the same. Now, imagine there's this evangelical organization back at home, which says, oh, these missionaries are doing a wonderful job, so we have to help them. So they pass the collection bowl around, and they collect some money, and we want to figure out how to help the missionaries. And so they kind of come to the wonderful idea, let's give the missionary cycles. That way they can quickly zip around the island, recruiting more missionaries to the cause. More cannibals to the cause. So, so that's what they do. The next boat to the island offloads these, very shiny cycles, and the missionaries are very happy. So they can zip around very fast, much faster than the cannibals can move around. And you think that everything would be hunky-dory, but suddenly after some time, you see that a qualitative transformation has taken place in the island's population. So suddenly you have regions which essentially are exclusively missionary rich, and regions which are cannibal rich. Okay, so from a situation where cannibals and missionaries were more or less homogeneously distributed around the beach, you suddenly have formation of patches such that there are patches where there are large number of missionaries, very few cannibals, and other patches where it's mostly cannibals, very few missionaries. So what happened? So what happened was introducing cycles in the system such that suddenly one population could move much faster than the other, and in particular the inhibitors could move much faster than the activator, introduced what we call an instability. So now, if at any point, if you have a small excess of cannibals, what would happen is that the few missionaries that you create initially from those excess cannibals are no longer sticking around to create more missionaries. So the moment they are, like, you pour holy water on them and you pronounce them, okay, so now forth you'll be known as John and you shall be a missionary. And here's your cycle. They gleefully take the cycle and zip away, right? They don't stick around to convert more of their brethren into missionary status. And so you have a system where regions which are, for whatever reason, noise or something, develop some kind of a small excess, keep producing more and more cannibals, and regions which are devoid of cannibals start getting more and more of these missionaries because they're zipping away from wherever they're created with their new cycles. So without the diffusion, these extra missionaries were reducing the cannibal excess, and so that the system could return to balanced state. But with diffusion, the missionary excess is transported away very quickly so that you actually maintain the excess cannibal in certain regions, and you have also created now regions having excess missionaries. So from a situation where there was no pattern, so to say, everywhere was same in the sense that you had the same proportion of missionaries and cannibals. You now have heterogeneity. You now have a breaking of symmetry because there are certain regions which are clearly distinguished from other regions, because in certain regions the proportion of missionaries to cannibals is much higher than in other regions. So what was earlier completely rotationally symmetric, if you think of the island, which is essentially just a circle, initially where there was complete rotational symmetry, you have actually a breaking of the rotational symmetry. Now this is very surprising, right? Because typically we think of diffusion as something which kills heterogeneity. You start from something which has extremely high concentration at a certain point. So for example, let's say you put some sugar into your tea. After a while, you expect your tea to be uniformly sweet, because that sugar which initially settled itself at the bottom of your cup would be diffusing and essentially taking over the entire cup. And yet here you have a system where diffusion is actually creating heterogeneity out of homogeneity. So this tells you about the non-trivial power of nonlinearity, the fact that you have a reaction which allows local activation, that when coupled to the inhibition, diffusion, actually allows diffusion or diffusive instability to actually create patterns out of nothing. And so if you actually look at what kind of patterns this mechanism creates, you see that it can create things like stripes. So black and white correspond to high concentration and low concentration respectively of the activated chemicals, or you could also have spots. So initially when Turing presented this idea, in the 60s and 70s, there was a lot of excitement among many people that maybe this could explain, for example, the stripes on the zebra or the spots on the cheetah. Unfortunately, it turned out that nobody could actually experimentally show the working of the Turing type self-organized pattern formation mechanism in any natural setting. I mean, let alone within an organism. People couldn't even actually experimentally show Turing patterns to form in any artificial system. So that was more or less the situation until, quite by lucky accident, I think in the late 80s or the early 90s, suddenly, people saw it in chemical experiments. And after that, of course, in 1995, for the first time, a Turing-type pattern formation mechanisms was shown to be operational in the marine angelfish. And later on, also the zebrafish was claimed to be showing, the Turing-like pattern formation mechanism, and a very characteristic, signature that it is in fact Turing-like pattern formation mechanism is that as the system size grows, the stripes actually increase in number. So, for example, here you see that as you go from a small fish to a large fish, the small fish essentially, which had, let's say, 1, 2, 3, 4, 5 stripes, essentially now has 6 stripes. This is very different, for example, in the case of mammals, like, for example, the zebra, where the pattern is actually laid out once and for all. As the zebra grows, it's not like it has more and more stripes actually forming in its body. It's just that the spacing between the stripes actually become larger. The number of stripes still remains the same. So, people have said that it's a bit like, you draw spots in a balloon or draw stripes in a balloon and you blow it up. The number of spots and number of stripes wouldn't increase. It's just that the interval between the spots or stripes keeps getting larger. So that way, the Turing pattern formation mechanism is very different because it argues that the interval between the stripes is always going to be maintained. So if you have a larger domain, you're going to have many more stripes. So that way, we can clearly distinguish a system which has something like a self-organized pattern formation mechanism operating vis-a-vis something which has, let's say, a boundary organized pattern formation mechanism operating. Now, of course, this idea that Turing had of lateral inhibition, that something, a particular cell, by promoting its own further activation, is going to, let's say, dampen the activation of its neighbors, is not just true for the particular kind of system that Turing was interested in. And lateral inhibition is, of course, far more general. And it operates across scale. So, for example, the orientation columns that you have in the visual cortex also very much, works on this lateral inhibition principle. It turns out that, the first person to kind of propose that something like lateral inhibition may be working in development was actually not Turing. So, at least 12 years before Turing actually published this paper, in 1940, the British entomologist Vincent Wigglesworth wrote this paper where he argued that the bristles that occur in the abdomen of various insects, so, he himself was particularly interested in this blood-sucking insect called Rhodinus prolixus, which is the vector of Chagas disease. So if you look at the abdomen of the larvae of this insect, you find these bristles, which are, you can see this very small, tiny circles located over here. So if you magnify, you find this pattern of bristles. So bristles are basically the sensory organs of the peripheral nervous system, which are essentially forming something which looks like a hexagonal close-pack structure. So they are very evenly spaced. So the question that Wigglesworth asked was, how does this even spacing happen? And so, unlike the classic tradition of British entomology up to that point of time, which just involved classifying insects into various genera and so on, Wigglesworth decided to do experiments. So he decided to, for example, burn out particular parts of the cuticle and see, how it would develop or how it will regenerate. And in doing this, he actually came upon this fascinating, realization that essentially each of the bristle-forming units, what are known as plaques, essentially exerts an inhibitory influence on the plaques which surround it. So each plaque, if it manages to, form a bristle, would essentially inhibit all of the plaques around it. And it does so by a process of substrate depletion. So essentially, you suck out whatever you need to develop bristles from your neighbors. So, you use up your, fuel, so to say, but you also use up those of your neighbors. You're basically stealing from your neighbors. And of course, every cell which kind of, forms a bristle is going to do that. It's going to steal from its neighbors. So now, if your neighbor tries to form a bristle, it won't have any juice to do it, right? Because you have already sucked away all that juice. So it won't be able to do the bristle formation. So only, a plaque which is far from you, and so therefore which still has its juice intact, which you have not been able to suck out, would still be able to form a bristle. And so therefore, you would have this is forming only at evenly spaced intervals across the abdomen of the insect. So this is a complement to the kind of activator inhibitor picture that was given by Turing. But the interesting thing is that both actually are reaction diffusion mechanisms. So Wigglesworth was unfortunately not trained in mathematics, so he didn't realize this connection. Although he lived till 1998, actually, he was extremely long-lived. He never actually connected his picture with that of Turing. But others, like Meinhardt in Germany, quickly connected these two together, and we realized that so reaction diffusion can actually manifest in two forms. You could have activity inhibitor of the type that Turing talked about, or you could have substrate depletion of the type that Wigglesworth talked about. So now that we have lateral inhibition as a clear important player in developing pattern formation, the question was, can you actually do it with, instead of the very simple dynamical elements that Turing talked about, can you do it with oscillators? So this is the idea that Brian Goodwin, who was another British developmental biologist, another pioneer in the field, started thinking about in the 70s. So he said that, look, in many biological systems, you see oscillations. In fact, if you start looking for oscillators in biological systems, you are going to find it everywhere. Oscillators exist in different spatial scales and different time scales, only a few of which are actually mentioned here. Moreover, the interaction between oscillators, allowing them to synchronize or not, actually plays a very, very important functional role. So, for example, the pacemaker cells in our hearts actually depend on synchronization to make sure that it kind of acts as a reliable timekeeper, such that we keep having our hearts getting contracted in this regular beat. Other examples are, for example, the beta cells in our pancreas, whose synchronized oscillatory activity allows insulin to be secreted. So if you actually do anything to disrupt the synchronized oscillation of these beta cells, it turns out that insulin will no longer be secreted. So this is responsible for certain kinds of diabetes, for example. So the beta cells could individually be perfectly fine, but if the communication between them is disrupted so that their activation is no longer synchronized, essentially they will stop functioning properly. You also have nonlinear synchronization of activity of neurons across the brain, which allows us to form certain sensory impressions. So without synchronization of these neurons, we simply won't be able to for example, visualize images. And of course, the contractions of the pregnant uterus, which allows a child to be born, those contractions essentially happen because the muscle activity across the uterus are coordinated. And this allows the periodic activation of the muscle cells to be synchronized. And of course, one of the most spectacular examples of synchronous firing of oscillatory activity is the flashing of fireflies in many Southeast Asian forests. So there's this wonderful episode in The Trials of Life by David Attenborough, where in one sequence, Attenborough actually takes a canoe and goes along the river of Southeast Asian forest. And you can see that as the film proceeds, so initially there are a few fireflies flashing together. Then eventually more and more fireflies would flash in synchrony, and eventually the whole tree lights up like a Christmas tree and then switches off. Lights up, switches off, lights up, switches off. It's completely spectacular. So what Goodwin was asking that, what would happen, or what actually allows these oscillators to kind of interact with each other so as to give rise to certain spatiotemporal patterns. In particular, can the kind of patterns that we see in development arise because of interaction between oscillators? And so the question that we started asking ourselves a few years back was that what happens if we allow oscillators, which would be realistic, for example, in a biological context, to have activatory inhibitory interactions with each other. Now, this, of course, was initially motivated by a set of very intriguing experiments done by Epstein's lab at Brandeis University, where they were taking oscillating chemical reactions. They were injecting this reactants in a microfluidic device. So there was oil in this channel, narrow channels, and they would inject this essentially water-based chemicals into this channel filled with oil. And so, we'll have pinching such that spontaneously these chemicals would form beads. So you would have a system where you have a chain such that you will have beads of chemicals separated by columns of oil. So you'll have bead, oil, bead, oil, bead, oil, and so on. Now, the oils are such that they allow diffusion of the inhibitory chemical, but not the activatory chemical. And so here you have an experimental system which actually exactly implements lateral inhibition Turing chemical oscillators. And what they found was that this particular system can exhibit a whole range of different patterns, including anti-phase synchronization of these oscillators, and also time invariant spatial patterns very similar to those which were seen by Turing. In fact, they had initially, when they saw these experimental patterns, they had argued that this must be Turing patterns because you have alternating beads arrested at a high concentration of a particular chemical, and the neighboring bead arrested at a low concentration of the chemical, and so it was high, low, high, low, high, low pattern, which is very characteristic of a Turing pattern. So, about decade back, we kind of got into this, trying to model this, and so my then student Rajiv Singh performed this very simple model where he said that, look, I can describe each of these chemical oscillators by using the well-known relaxation oscillator model, namely the Developmental model, and I could couple them together using the inactivation variable. So essentially, we are positing that the inactivation variable is diffusing across the system. Now, what we saw was that we could actually reproduce all the experimental patterns and then some. So we could see the synchronized oscillations, we could see the anti-phase synchronization, we could see what the experimenters called Turing patterns, but what we realized was not really Turing patterns because they don't actually obey the conditions of Turing pattern formation. But of course, visually they're very similar because neighboring beads would actually be arrested in high, low kind of concentration states. But what is happening is that the diffusion is actually causing the arrest of the oscillations, and the arrest is happening such that neighboring oscillators would be arrested at dissimilar states. So we called it the spatially patterned or heterogeneous oscillator death state. But we also saw other kinds of patterns, which experimenters actually verified after we published this paper, which we called the chimera state because essentially there were coexistence of different kinds of patterns. So they had coexistence, for example, of time invariant patterns like the spatially patterned oscillator depth with the synchronized oscillations. So by varying the parameters of the model, we could actually look at a variety of different patterns, including that of moving defects. So defects would be essentially any disruption in the overall pattern. So for example, if you have complete synchronized oscillation, and at some point you have, let's say, a cell which is not oscillating, that would be a defect. Now, if it doesn't move, then you have, what I'm showing here. But if it starts moving, then you have, beautiful, patterns which are formed by these defects actually gradually moving, in a kind of a ballistic trajectory. They can collide with other defects. The collision can lead to other kinds of patterns, or they can just pass through each other. A whole bestiary of different patterns can be seen. When you look at it in 2D, you find even more spectacular patterns. For example, spiral waves. And under certain conditions, the spiral waves can actually form on a checkerboard pattern, which are oscillating on or off. Or you could have completely frozen pattern where different cells are arrested at different states. So you have something like a checkerboard, but with kind of beautiful spatial patterns. And so we started wondering, is there a possibility that this could be seen in a biological context? And we chanced upon a very well-known model of ecological population dynamics, which essentially talks about what would happen if you have a predator population and a prey population interacting with each other. So of course we are very familiar with the Wolpert model, those of us who come from a knowledge dynamics background, but unfortunately if you talk to an ecologist and you just happen to mention Wolpert, that's the last you'll ever hear of the ecologist. However, Rosenzweig-McArthur model is something that an ecologist is also comfortable with. So, we took this Rosenzweig-McArthur predator-prey model, and what we said was that, look, there are many systems in which the prey, for example, vegetation, does not move. So there's no question of it moving about. But the predator could have locomotion, so it could actually move around grazing on the vegetation. So suppose at the first approximation, I model this locomotion as some kind of a diffusion, just random movement. And I could have a diffusive mechanism where I have diffusion in the inhibitor, because the predator is in this context the inhibitor, and no diffusion in the activator. And lo and behold, we found that we can get exactly the same kind of patterns that we saw in the case of the chemical model. And we could see spiral waves, we could see spatially patterned oscillator death. And surprise, guys, we found that similar reports have been made of patterns in ecological context. However, there is a problem when we start talking about applying this in the developmental context. Well, for one, diffusion is typically too slow, because you have large length scales, related to which diffusion is actually not very good at communicating across large expanses, or it could actually be insignificant because of cell wall. So the stuff that is inside a cell simply can't get out because there is a cellular boundary, the lipid bilayer, which prevents straightforward transport from one cell to the other. And so the question was, could the kind of patterns that we're seeing arise if we incorporate biologically realistic interaction mechanism between cells? And so, right about 3 or 4 years ago, we started looking at this question that what would happen if you have cellular oscillators which are interacting with each other via receptor-ligand interactions, which are essentially implementing this lateral inhibition. So an example of such a developmental pattern which arises through precisely this kind of interaction is somatogenesis. So I briefly alluded to somatogenesis when I showed you the zebrafish embryonic development video. So here are a few stills from a similar video. So you'd notice that as the zebrafish develops, its backbone, what would eventually become its backbone, starts developing these notches. So these are what essentially would form the vertebrae. And what you notice is that the tail, as it gradually expands, so this is the tail part is known as the presomatic mesoderm. So this is what eventually develops into somites. So as time progresses, more and more somites form at the end of this chain of somites. So over time, we go from 12 somites to 15 to 18, 21 to 24. And what we know from experiments is that prior to the formation of the somites, each of the cells is undergoing oscillations where there are certain genes known as segmentation clock genes whose expression basically shows an oscillatory variation, but which at the time of formation of somites gets arrested, and the phase at which the oscillations get arrested essentially decides whether it will form a somite or a somite boundary. And so this is how this very characteristic, notch-like somites tend to get formed. So in 1976, Cook and Zeeman, so this is the same Zeeman who was, instrumental in promoting catastrophe theory back in the 70s, argued that there could be some kind of a generic mechanism at work, which they call the clock and wait fund scheme. So the idea was that each of these cells, which is undergoing this, oscillatory gene expression, could be thought of as a clock. So initially, suppose, all of them are synchronized in their beating. So all the clocks are essentially synchronized. But then you have a wave of some type, which is emanating from one end. And as soon as a clock meets the wave, it immediately gets frozen at whatever phase it was. And so you can imagine that if you have a wave which is progressing across the array of oscillators at some specific speed, it will arrest the various clocks at different times or different phases, so to say. Of course, this is probably a classic prop in all detective movies, right? Exactly at the time that a person was shot to death, the clock stopped because the bullet happened to hit the clock. Okay? So just imagine, you have several such grandfather clocks, and you let's say fire a gun or something, and every time the bullet goes to a particular clock, it's freezing the clock at whatever time it was, at that point of time. Now, of course, bullet travels very fast. So imagine that these clocks are separated very wide apart so that the bullet, as it enters each clock and arrests it, the clock is going to show a different point of time. And so by looking at the clock after the incident, you can actually see the clocks all frozen at different points of their circular motion. And so the idea was that a particular phase corresponded to being a somite, and the other phases corresponded to being a somite boundary. So beautiful idea, except that there was no details. There was no concrete scheme of what exactly is the wave, how are the oscillations arrested, and is there some way, some kind of a unified mechanism by which we can describe the entire process of somatogenesis. Now, this becomes a particular challenge because actually there are three different spatial scales at which you can try to understand somatogenesis. There is the cellular scale, where you're trying to understand the single cell oscillators, whose genetic circuits are actually determining the period of the clock. Then you have the intercellular scale, where the coupling between the cells are what allows the different cells to synchronize the oscillations. And finally, you have the pre-somatic mesoderm scale, the scale of the entire tail, at which the mechanisms by which the cells arrest their oscillations, forming the halves, the rostral and caudal halves of the somites, are operating. So how do we actually go about trying to tame this beast, so to say? So this is a schematic of the zebrafish pre-somatic mesoderm. So the important point is you not only have clocks working here, you also have a morphogen in the sense of Wolpert, forming a gradient. So there's a chemical which essentially has a concentration gradient and which therefore in principle allows every cell on the pre-somatic mesoderm to figure out where it is relative to the entire system. It has a sense of its relative location within the organism. So what we want to do is basically come up with a mechanism such that you would have a spatiotemporal progression from synchronization, which occurs at the tail of the organism. Now remember, the tail itself is not static, but it's essentially just progressing more and more towards the right. So more and more cells are being added, and the tail is progressively going further and further to the right. So the cells which were originally forming the tail are, after some time, no longer the tail. They're somewhere in the middle of the pre-somatic mesoderm. So what you should see is that synchronized oscillations at the tail, which kind of give rise to traveling waves somewhere further. Then eventually these oscillations will stop. And after some time, you should see the formation of somites. So in terms of nonlinear dynamics, what you should expect are exact synchronization at the end, at the posterior end. Then traveling waves essentially correspond to a gradient of synchronized states, so what we call gradient synchronization, or GS, at this region. Homogeneous steady state corresponding to oscillation arrest over here. And finally, at the anterior end, you should have a inhomogeneous steady state corresponding to different cells or alternating cells arrested at high state and low state. So what is the interaction mechanism that we're using here? So as I said, diffusion doesn't work. So we used what is known as notch delta signaling pathway. So notch is essentially a receptor which is located at the surface of a cell. And delta is a ligand, a protein, which essentially, when it exactly sees a matching notch, will lock onto it. And this will result in a signal being transmitted to the cell whose notch receptor has connected to the.
So Sinha, I'm sorry to interrupt. I'm really sorry, but maybe we can wrap it up in 5-6 minutes.
Sure. Yeah, absolutely. Yeah, I'll just do it.
We can take some questions also then.
Sure.
Yeah.
So this actually can be formulated in a very detailed model of notch delta signaling, and the oscillator within each cell is of course driven by genes regulating each other's activity. So that forms a kind of relaxation oscillator. So here you have a set of equations which couple the genetic oscillators of one cell with the genetic oscillators of neighboring cells via this kind of a notch delta coupling. And we find that for a given strength of coupling, so the coupling is essentially governed by these parameters F, G, and H. So if you fix F, G, and H, you would see a certain kind of dynamical pattern. So depending upon what is the value of F, G, and H, you could see either exact synchronization, or you'll see gradient synchronization, or you'll see homogeneous steady state, or you'll see homogeneous steady state. So separately, you will see each of these patterns that are seen at different stages of somatogenesis. So the question is, then how does it actually work out such that the same cell at different points of time is going to go through each of the stages? And so this is where a morphogen gradient becomes important, because that actually provides you the information about where you are on the presomatic wisdom related to the tail. So if you're very close to the tail, you should do exact synchronization. But if you're very far away from the tail, you should actually form the somites. So we give, and of course, there is experimental evidence of such morphogens. So for example, retinoic acid is a particular chemical which clearly fits the bill for the kind of morphogen that we are presenting. So once you have such a source of morphogen, it forms this exponential gradient. And when you look at how it operates on just a pair of neighboring cells, you find that this different concentrations of the retinoic acid seen by the two cells essentially results in a sequence of dynamical transitions by which they go from synchronized oscillation to oscillation arrest to diverging steady states. So one cell goes on to become arrested at a high concentration, the other cell goes on to be arrested at low concentration. So essentially they form the rostral and caudal halves of the somite. So what you can show for a pair of cells, you can also show for an entire array of cells. And sure enough, we find that if you take an array of, for example, 20 cells, initially when the morphogen concentration is at a particular level, you find waves of periodic activity, which essentially eventually becomes traveling waves, then leads to oscillation arrest. And finally, you have something which looks like Turing, but is actually just oscillation arrest, which corresponds to the formation of the somites, the rostral and caudal halves of the somites. So what we have here is a model that essentially not only explains all the key transitions that you see in somatogenesis, but more importantly, it provides a framework that allows us to combine two otherwise very distinct symmetry breaking mechanisms. So on the one hand, you have the self-organized mechanisms, which just uses local interactions. And on the other, you have the boundary organized pattern formation mechanism, which uses a global field, namely the morphogen concentration gradient. So typically, they used to be thought of as very separate, pattern formation mechanisms. So, if you apply global fields to form patterns, you cannot use local interactions and vice versa. But here we show how you can actually fruitfully use both of these types of mechanisms to actually explain all the known or all the observed features of a particular pattern formation phenomenon that is seen in nature. So I'd like to end by thanking the various people who have been involved in this work. So Rajiv was instrumental in actually forming the fundamental theoretical basis for understanding how patterns can form by having oscillators interact with each other through lateral inhibition. So he's of course now active member of the ITBHU. Chandrasekhar, who's just finishing off and writing his PhD thesis at the moment, is the one who actually extended this to understand developmental panel formation. And Shakti Menon, who's a project scientist at the Institute of Mathematical Sciences, has been very much instrumental in developing these ideas, not only to explain biological phenomena, but also to see whether things like, for example, can we do chemical computation using this paradigm of lateral inhibition between relaxation oscillators. So I'm sorry for going out of designated time, but I'd be happy to answer any questions.
Thank you so much, Professor Sinha. That was a very engaging talk on the secret of pattern formation in biological systems. We can quickly take some questions. Participants, you can type your questions in the chat box or can raise your hand and I can unmute you. Are there any questions?